Netherlands. I am very pleased about this day already. For the people that haven't been here in the afternoon, we had a fantastic program with the EIT ICT Labs startups. We had fantastic presentations from local Silicon Valley startups. A lot of discussion, I really enjoyed it. We had a fantastic pitch training of the fruits of which we will see later tonight. Just very briefly, we are here at the Consulate General of the Netherlands. My name is Robert Thijssen. I am the head of the Netherlands Office for Science and Technology, which is one of the teams that we have here at the Consulate. We work on connecting Dutch and US science, technology and innovation communities. We have a broader team of economic people working here in the economic department, working on trade, connecting businesses from the Netherlands and the US. We have also people trying to attract, very successfully I have to say, foreign investment to the Netherlands. And I'm very grateful that we could partner tonight with EIT ICT Labs. And Bay Area Life Tech. Luke Lightning over here. Just some practicalities, probably all already know, but the restrooms are outside. We have food. I guess most of you had some. We have drinks. Please take it. I'm going to round up here. I want to.
They have to-go cups.
I'm going to give the floor to Willem Jonker shortly. He is the director of the EIT ICT Labs. Then I will hand over for the rest of the night the moderation to Luke Lightning of Bay Area Life Tech. And so the program is we will have a presentation by Adam. Hi. We will have 6 pitches. 2-minute pitches of European startups. We will have a break. We will have a presentation from Rock Health, the leading health IT, or I should say digital health incubator in San Francisco. And then we will have another 5 pitches. And here I'm going to stop. And I would like to give the floor to Willem to give some words about EIT ICT Labs. Thank you. Enjoy.
Robert, thanks very much. First of all, of course, for hosting us here today. And we're really very happy with that, and also all the efforts that you put in getting this co-organized with it. They said, Willem, you only get 5 minutes, and this was very a relief for me, because several times this week they told me, it's only 5 minutes away. So you know how long this talk will last, right? So, what is EIT ICT Labs? EIT ICT Labs is an innovation organization that really focuses on ICT innovation and education, with the mission to make sure that the European ICT landscape is really world competitive. And we do that in a private partnership setting where we are financially supported from the European Commission, but also from national governments, and they bring in around 25% of our budget, and the rest we bring in through our partnership. We operate from 9 countries in Europe, and we really are trying to have the key ICT players in the different countries in our partner network. We have about 100 partners. It's a very young organization. We started operations in 2011. We were starting with a relatively modest, let's say, first budget of around 50 million euro in the first year. And we're scaling up very rapidly. So for next year, we just completed the business plan, and we will be on a run rate of 300 million. So the organization is scaling up significantly. The reason why we are here is because we now have, let's say, the base organization in Europe operational, functioning, and producing results. And we feel it's very important now to connect this European cluster of ecosystems to another very important ecosystem, which is Silicon Valley. And what we are running today is one of our first pilots where we want to get a feeling for the kind of relationships that we can successfully build between technology being developed in our ecosystem network in Europe and the community here in Silicon Valley. And that's why we brought a collection of initiatives here. They will pitch to you, and what we want to find out, very simply, is this of interest to you? And of interest does not mean the Japanese way of saying, very interesting, thank you sir, but more the Dutch business way of interest, if you know what I mean. So, I hope you will enjoy it. I will definitely enjoy it, because I can just sit and relax. And I hope that this will help us to better shape our interaction with the Valley, and to also guide us in building our ambition of really to connect to the Valley, our ecosystem, and the local ecosystem here. Thanks very much for coming, and please enjoy. Thank you.
All right, welcome everybody.
Oops.
I was afraid of it.
Excellent way to start. Show off my dance moves right there. How about that? Thank you. Holy cow. Anybody that's pitching, don't do that.
It won't happen again.
Welcome everybody. My name is Luke Lightning. I am the founder of Bay Area Life Tech, an organization now that has over almost 1,100 members. We started about a year and a half ago, and now we're growing at a rate of about 100 members per month, which is awesome. I'm really excited about that. Some people in the room tonight asked me what I actually do for a living. So just briefly, I will tell you that my background is that on? Okay, my background is in biotech. I have a PhD in chemistry. I was involved in an organization that raised over $300 million, went through an IPO about 3 years ago. And now I've transferred myself and my skills, some of my skills anyway, over into the technology space, and I do business development for a company called Launch, which is based here in San Francisco and in LA. We do big events for technology startup companies to get the word out. Exactly what we're going to see tonight in the biotech and life tech space. It's nice to see companies like Beddit overlapping. Beddit actually presented at our conference here 2 days ago. So it's really exciting to see the overlap. I'd also like to point out that any startups that are here that are pitching, we have a blogger for Bay Area Life Tech. She's sitting here in the second row, Karen Ring. She blogs about all our events, does recaps. So if you'd like to give a short interview, she can recap that for you, and hopefully it will make it into publication press in a few weeks. So with that, oh, by the way, I'm really happy to, we're a United States-based company, but I'm very happy to partner with a European company, especially tonight, given that the US Consulate is probably shut down. Thank you, Europe. Thank you, Europe. All right, so without further ado, I'm going to introduce Adam Mendelson, our featured speaker tonight. Adam actually has been a Bay Area Life Tech member almost since it began a year and a half ago. I looked that up today. I was like, wow, that's awesome. So I'm happy to see him here, and I'm happy to introduce him as our featured speaker tonight. And if anybody that is a member or anybody outside the group, obviously, would like to speak or present or have something interesting to present, please send me an email. I'm pretty accessible on the website, and we can talk about it. So we like to highlight all of our members. So Dr. Adam Mendelson, who I've gotten to know very well. He's also on the board at UCSF for the alumni group. He's our featured speaker. He serves as the CEO of Nano Precision Medical, which is a medical device company, which he co-founded in 2009 to develop a subdermal implant capable of precisely delivering therapeutic molecules precisely? Oh, really precisely? How precise? Very, okay, good. Over extended durations to improve the treatment of chronic diseases. He's got a fantastic background from UCSF, and I will hand it now over to Adam. Thank you.
Well, thank you, Luke. Watch your step.
Yeah.
For the first event that I went to was here, and I had a wonderful time. So I'm glad to be back, and I'm glad to have all of you here. Thank you to the organizers. Thank you to Luke. I want to start by saying 2009 is when we started the company, but it was in 2007 that we first started to form our business plan pitch. And the first pitch that we gave was a 7-minute pitch as part of the UC Berkeley Nano Club. One of my co-founders was the president of that club at the time. And they were desperate to have people enter the competition. And my other co-founder, Lily, and myself and Kate, were all working late one night in the lab at UCSF. And Kate was, again, in her desperation, trying to get people to enter, convinced us to do that with her, and that was the beginning of Nano Precision Medical. But the first pitch that we gave was almost entirely about the technology, because as scientists, as I think many of you are, we love technology. We love talking about the science and the technology, and it's what drives us, and it's what we are passionate about. But then in 2008, by winning that competition at the Berkeley Nano Club, we were placed into the semifinals of the UC Berkeley Business Plan competition. We were under a different name at the time, Titan Medical. It turns out that somebody else had that name, so when we went to form the company, we had to find one that hadn't already been taken. But we had to write a full business plan. We learned about markets. We learned about how to give a pitch. We were assigned mentors. We ultimately came up with a business plan presentation that had almost nothing about technology in it. And we talked a lot about the market, a lot about the regulatory issues, a lot about the financials and how successful the business could be to provide a return on investment for the investors. But the technology had basically one slide: yes, it can work. And that's all a lot of the people that we were speaking with seemed to be interested in. So I wanted to preface what I'm going to present, because this is not a business plan pitch. There's a lot of information that we would provide in such a pitch that is not here. And because it's not a business plan pitch, I'm going to talk about the technology, because that's what I'm interested in again. So this is back full circle. And of course, if you have any questions, I'd be happy to answer them. But without further ado, let's get started. My name is Adam Mendelsohn, as Luke Lightning mentioned, and I am one of 3 founders and the CEO of Nano Precision Medical. We are developing a small implantable device, and by small, I mean about 2 millimeters in diameter, that will go underneath the skin and provide long-term release of small and large molecule therapeutics. We see this device having greater opportunities as newer molecules that are being developed have higher potencies, and therefore can enable longer-term release of medicine in a small enough device that is meaningful, that can be meaningful to patients and will be clinically useful. Now, what I'm about to show you here, if this works. Okay, maybe I'll use this. So, the goal that we are trying to achieve is to maintain the levels of a drug at a constant state over an extended period of time. And this can be particularly useful in a variety of chronic disease treatments where people need frequent injections in order to maintain therapeutic concentrations. Right now, we're focusing our efforts on a peptide for type 2 diabetes, for which there is already data that exists suggesting that constant levels may even be more effective than the injections that are currently being used. But we're always looking at new molecules, new opportunities, and if anybody has a high potent molecule for which constant rate delivery could be useful, we have a technology that we think could be good to apply to delivery of that molecule. Now, the way the technology works, and we started out by talking about movie theater analogies and and hourglass analogies, because it's difficult for people sometimes to comprehend that without any moving parts and without any electronics, that we can simply maintain the release rate of molecules at a constant level over an extended period of time. But this is literally hot off the press, except it's not working. Oh, there it is. So standard, so this is actually three-dimensionally rendered. This is an actual animation that's created from real physical spheres, thousands of them inside a tube. And in this tube, you can see that the opening for all of the spheres is quite large. Many, many can fit at a time. And under this scenario, standard Fickian diffusion, where the concentration gradient determines the rate at which molecules escape from the device, governs the release. This is what you would expect based on standard science. And you can see, as we let this simulation play out, you can see that the rate at the beginning is quite fast, and as the concentration inside the device becomes lower, then the rate becomes lower as well. And this continues until there's almost nothing left inside the reservoir, at which point you have very little release. And this is a standard membrane with standard diffusion. Now this is not ideal for an implantable device for which we want to maintain plasma levels. Now, what becomes interesting is if you have a pore size that is about the same size as a molecule, you can see that it's actually quite slow at the beginning. And even as the concentration decreases on the inside, it maintains its rate for a considerable portion of its delivery. Now this has been observed by multiple different organizations since the beginning of the 2000s in a variety of materials. The materials that had been used initially were not ideally suited for biocompatibility purposes. Our company was founded based on the ability to achieve such precise pore sizes using titanium oxide, which has a long history of biocompatibility in humans. You're going to see some images shortly. But first you'll see some data. This is the only data that I'm showing today. There's more behind this if anybody has any questions later. But with the same size membrane, with a smaller molecule versus a larger molecule, you can see they're both quite sustained, but the true zero order constant rate release is achieved when the molecule size approximates the size of an individual pore. And this is the law of nature that we're exploiting in order to be able to, without any moving parts again, with one material and only with titanium, which is quite biocompatible, to be able to achieve this goal of long-term constant rate release. This is what the nanotubes look like. I'll do a short aside because somebody mentioned about the Japanese culture. And this is also an aside for people that are starting companies, particularly from universities, is when we went to incorporate in December of 2009, the reason we did that was so that we could have an entity to own a technology license. The technology that we were initially negotiating for was owned by a university, but it was a patent application. It had not yet issued. And the first thing that we did, and the best money that I still think we have ever spent, was hiring an intellectual property firm to do diligence on the patent application to make sure that those claims were going to issue. As a result of that diligence, we realized two things. One, there was prior art that both would make it highly unlikely that those claims would issue, and two, that prior art were pre-existing patents that we needed. So we did two things. One, we chose not to license the university patent application, which, by the way, has since been abandoned because the examiner found the prior art that we knew that they were going to find. And two, we went and sought an exclusive field of use license to use the technology that had already existed. Now, this technology was developed by a Japanese company, and over the course of about a year and a half, with not knowing whether they said they were interested in licensing to us, whether it was just Japanese culture, I was up late at night, 2, 3 in the morning, checking emails because that's their business hours. But ultimately, we were able to get signed contracts, and they've honored them ever since. And that's the other thing that I've noticed about doing business with Japanese companies, is that they honor their contracts very, very well. And it was as a result of building the team that we had, as well as having some solid intellectual property to protect ourselves from other people using the same method to do what we're doing. That's why we were able to raise funding, in my opinion, if I look at the mindset of the investors. And so in this kind of business where it's a long time scale before you have a product, having strong intellectual property at the beginning is highly important. But this is what the nanotubes look like. They're actually, we can grow them quite long and straight. They're very precise with regards to the diameters. They're actually different diameters on the bottom than they are at the top. And based on how we grow them, it's an electrochemical anodization process that we use. We can have very good control over what those pore sizes are, over what the length of the tubes are, and because they're highly ordered, it allows this to be used as a membrane made from titanium oxide with very precise pore size control to use for drug delivery. Now what we have done since is we've taken this membrane and we've left it attached to titanium, which is used ultimately to cap a titanium reservoir such that the only material is titanium and titanium oxide. And we can selectively decide what areas of the titanium membrane we would like to have open, essentially the number of nanotubes that are going to be accessible to drug molecules, which correlates with the rate of release. And if you zoom in, and I have one of these membranes with me if anybody wants to take a look, if you zoom in on one of the windows in the titanium, and you zoom in on the window far enough, you see an electron micrograph of what the uniformly opened nanotube bottoms look like. And again, we have control over this, and it's the same across the entire window area. Now, this technology is really a platform technology. We can control what the delivery rate is going to be simply by controlling the opened areas through which the nanotubes are accessible. And we can accommodate a variety of molecule sizes by adjusting the small end of the nanotube and what that diameter is to approximate the size of the molecule that we would like to deliver. And lastly, this is how the whole thing comes together. You have lots of drugs inside the reservoir that are for which the size approximates that of the pore size of the nanotube. The nanotube sits onto a patterned titanium disc, which caps the reservoir, and ultimately we can have, these dimensions are actually quite arbitrary. It ultimately is a function of how potent the molecule is and what a clinically meaningful duration of implant would be for a particular patient population. But there's very little space that's taken up by anything other than the drug formulation, which is a little bit of a differentiator to some of our competitors. We're not the only ones doing this, but we are the only ones that are doing this with only titanium and titanium oxide, and we think we have a big advantage with regards to regulatory and marketing considerations as a result. We've looked at all of the molecules that are marketed and many that are still in development as potential candidates for delivery from our device. We've started a program, as I mentioned, around a type 2 diabetes peptide. There's others that we're thinking of starting, and we're highly interested in identifying additional molecules that could be interesting. But suffice it to say, we think that there's a large opportunity to apply this technology, and we think that this opportunity is only going to increase with time as molecules get more potent and are more targeted and more selective, and smaller amounts can be effective. And those are all the slides I have. I would be happy to take any questions. Luke told me I was only going to speak for 10 minutes, although I think I've already spoken longer.
No, that was great. That was great. Good job.
Great.
Let's thank Adam.